Hello, my name is Morgen Henry. I'm a research technician of the Veenema Lab at Michigan State University. Today I'll be discussing my recent characterization of oxytocin receptor binding density in the OXTR-iCre rat line targeting brain regions associated with social behavior. Our lab studies how neuropeptides, such as oxytocin, regulate various social behaviors through the activation of their associated receptors in the brain. Currently, little is known about the neural pathways by which the activation of oxytocin receptors regulate these behaviors. To study this, we have generated an OXTR-iCre rat line. OXTR-iCre is a transgenic line of rats that have had Cre inserted into all cells that express oxytocin receptors. These rats allow us to use Cre-dependent chemogenetic, optogenetic, and viral tools that help us study the functional and anatomical properties of oxytocin receptor expressing cells within the brain. These rats are a necessary tool for achieving our main research goal of understanding the role of oxytocin receptor expressing neurons and the neural pathways in which they function within the context of social behaviors. To do this, we must first validate the OXTR-iCre rat model and characterize their underlying neurobiology. Previously, we have confirmed their functional expression of Cre. Through this characterization, we hope to verify that the insertion of Cre does not alter the expression of oxytocin receptors. Our selection of brain regions for this characterization was determined through their association with social behavior, expression of oxytocin receptors, and existing age and sex differences in oxytocin receptor binding density. The regions we have chosen are the nucleus accumbens, bed nucleus of the stria terminalis, ventromedial hypothalamus, and medial amygdala. If the insertion of Cre has no effect on the expression of oxytocin receptors, we expect to find similar oxytocin receptor binding density between OXTR-iCre and wild-type rats of the same sex and age. We also expect to find similar age and sex differences in binding density within these lines. To perform this characterization, we utilized receptor autoradiography. This technique entails the application of a radioactive ligand that is labeled with I-125 and selectively binds to oxytocin receptors. On the figures, I've highlighted an example brain region, the nucleus accumbens, on both the resulting audiogram and the matching brain atlas figure. When film is exposed to this tissue, it is ionized by the radioactive ligand and a darker hue develops on the resulting autoradiogram. Areas in which there is a greater density of ligand bound to oxytocin receptors will appear darker. The following results are a comparison of the observed binding density of both sex and age groups of each genotype. In the nucleus accumbens, the bed nucleus of the stria terminalis, the anterior and posterior ventromedial hypothalamus, and the posterior dorsal medial amygdala, we found that OXTR-iCre rats have lower oxytocin receptor binding density than wild types. Here, all significant genotype effects are indicated through significance bars. In the nucleus accumbens, bed nucleus of the stria terminalis, and posterior dorsal medial amygdala, genotype effects are restricted to one age and/or sex. We were able to confirm previously found sex effects in the bed nucleus of the stria terminalis, the anterior ventromedial hypothalamus, and posterior dorsal medial amygdala. However, we found an unexpected effect of sex in the nucleus accumbens where males had higher oxytocin receptor binding density than females. This seems to be driven by our juvenile male wild-type group. We had also expected to find sex effects in the posterior ventromedial hypothalamus and posterior ventral medial amygdala, but these were not present in our findings. We were able to confirm previously found age effects in the nucleus accumbens, bed nucleus of the stria terminalis, anterior and posterior ventromedial hypothalamus, and posterior ventral medial amygdala. However, we had expected to find age effects in the posterior dorsal medial amygdala, but this was not present in our findings. Contrary to our hypothesis, OXTR-iCre rats had lower oxytocin receptor binding density in 5 out of 6 brain regions. This was restricted to one age and/or sex in 3 regions. From this, we can conclude that the insertion of Cre in the OXTR-iCre line may result in lower oxytocin receptor binding density, but this is inconsistent across age and sex. While we confirmed similar age and sex differences in the bed nucleus of the stria terminalis, we did not find our expected age and sex differences in the nucleus accumbens, ventromedial hypothalamus, or medial amygdala. Since we could not confirm previous findings through this characterization, it suggests we may have differences in our experimental design that produce different results. In the future, we plan to use qPCR to investigate if gene expression of oxytocin receptors differs between these lines, and Western blotting to investigate if cell viability of oxytocin receptor expressing cells differs between these lines. Depending on the results of these future investigations, We will adapt our statistical interpretation of future experiments to account for the underlying differences between these lines. We will also expand this characterization to include more brain regions that are rich in oxytocin receptor expressing neurons and are associated with social behavior.